You wake up, you try to swallow, and it basically feels like you've just ingested crushed glass.
Oh, the worst feeling.
Right. So you drag yourself to the bathroom mirror, shine your phone flashlight into your mouth, and yep, there they are, those telltale white patches on the
Just
tonsils.
throat.
Strep throat again. You probably had it in first grade, maybe again in high school, and here it is ruining your weekend as an adult. And, it really defies a basic biological expectation.
And we're taught that our immune system learns.
Right. Exactly. If you get the measles once or you get the vaccine, your immune system essentially files that mugshot away. You're generally immune for life. So why does this one, microscopic throat bug get a free pass to infect you over and over and over again?
It really feels like a glitch in our biological software.
Mm-hmm.
With this particular organism, it's like our immune memory effectively resets to 0 every single time.
And that biological amnesia is the focus of our deep dive today.
Right.
We're looking at one single organism, Streptococcus pyogenes.
Right. Universally known in clinics as Group A Strep, or GAS.
Yeah, GAS. And our mission today is to figure out how this specific bacterium can cause everything from, a mild, annoying sore throat to catastrophic multi-organ failure within 24 hours.
It's quite the spectrum, and honestly, the name itself is a bit of a spoiler alert for what it does.
Oh, yeah. The nomenclature is brutal. So Streptococcus comes from Greek roots.
Yeah, streptos meaning twisted, and coccus meaning berry.
Right, because under a microscope, they don't cluster in clumps like staph does. They grow in these long, twisted chains of spherical shapes.
Exactly, and the pyogenes part.
Yeah.
That literally translates to pus-producing.
Pus producing, just not a title you want on your resume.
Definitely not.
And in the lab, its microbial profile is super specific. It's gram-positive, catalase negative, and it has this trait called being beta hemolytic. And when I was reading the source material about how they test for this, it struck me as basically a microbiological crime scene.
That's a great way to put it, actually.
Yeah. Because if you swab a throat and grow the sample on a blood agar plate, which is literally a jelly made with sheep's blood, this bacterium doesn't just sit there on top.
No, it goes to work.
Right. It completely obliterates the red blood cells around it, leaving this perfectly clear, transparent halo.
Yeah, that clear halo is a visual readout of the bacteria's destructiveness. They produce these enzymes called streptolysins, and they basically puncture the membranes of the red blood cells to harvest their nutrients.
Specifically the iron.
Yes, exactly. And that beta hemolytic clearing, that halo, is the very first thing a microbiologist looks for to say, yes, a pathogen is definitely present here.
But pinning down the actual identity of that pathogen took medicine a frustratingly long time.
Oh, centuries.
I mean, you can find clinical descriptions of scarlet fever and erysipelas, which is this severe skin infection in ancient and medieval texts. But for the longest time, doctors had absolutely no idea that the bug causing a scraped knee to get infected was the exact same bug causing deadly postpartum sepsis.
Which they called childbed fever back then.
Right. Visually, I guess the pus looked the same, but the diseases were just so radically different that nobody connected the dots.
Well, the clinical presentation was just too varied. I mean, by 1884, a German surgeon named Friedrich Julius Rosenbach officially gave Streptococcus pyogenes its name. But even for decades after that,
It was still a mess,
a
right?
complete taxonomic mess.
The genus Streptococcus had dozens of strains causing distinct illnesses, and clinicians had absolutely no reliable way to sort them out, let alone track outbreaks.
Because, they couldn't just sequence the DNA back then.
Exactly, they were flying blind.
Until the 1920s and 30s. That's when a bacteriologist named Rebecca Lansfield at the Rockefeller Institute completely changed the game.
Her work was revolutionary.
It's fascinating because her methodology was pure brute force biochemistry. She basically realized you couldn't identify these bugs by how they looked, so she boiled them in hot hydrochloric acid.
Yes, quite literally boiling off the outer layers.
Just stripping them down to isolate the specific carbohydrate structures hidden deep inside their cell walls. And that approach changed everything because she moved the classification from the macro level down to the molecular level.
Right.
Once she extracted those distinct carbohydrates, she injected them into rabbits to see which specific antibodies the rabbits would produce. And using those antibodies, she sorted this whole chaotic family of bacteria into neat alphabetical groups.
Groups A through V.
Exactly. And Group A turned out to be the smoking gun.
It aligned perfectly with Streptococcus pyogenes.
It did. It definitively linked the sore throats, the scarlet fever, and the postpartum infections to one common enemy. It was a massive paradigm shift. She provided a universal language for infectious disease.
And what's wild to me is that, almost a century later, her work isn't just some historical footnote. When you take your kid to the pediatrician today and they run a rapid strep test, that little test strip is chemically hunting for the exact same Lancefield group A carbohydrate she identified in 1933.
We are entirely reliant on her framework today, and honestly, the speed of that rapid test is vital because symptom-based diagnosis for this pathogen is notoriously flawed.
I am so glad you brought that up, because there is this really pervasive myth that you can just look at a throat and know it's strep.
Oh, everybody thinks they can.
Right. People think, oh, if I have a fever and those gross white exudates on my tonsils, it's a guaranteed positive. Even doctors use scoring systems, like the center criteria.
Yeah, the center criteria.
Where you get a point for a fever, a point for swollen lymph nodes, a point for lacking POF.
Right, but while the center criteria are useful for statistical probability, they fail constantly in practice.
How so?
Well, a virus like adenovirus or Epstein-Barr.
Which causes mono.
Exactly. They can perfectly mimic the white spots, the fever, the severe pain, all of it. And conversely, a patient can present with just a mildly red throat, no white patches at all, and test strongly positive for group A strep.
Wow. So you really cannot eyeball this pathogen.
You absolutely cannot, which is why microbiological confirmation, whether that's a rapid antigen test or a throat culture, remains the gold standard.
Okay, so the rapid test easily spots Lancefield's carbohydrate shell. But that brings us right back to our opening mystery. If our modern lab tests can spot this structural fingerprint in 5 minutes, why is our immune system so completely blind to it year after year?
It implies there's something else covering up that carbohydrate.
Right. It's hiding.
It is. Group A strep possesses a staggering array of virulence factors, but the crown jewel of its evasive arsenal is the M protein.
The M protein.
Yes. It's a coiled coil protein encoded by the M gene, and it projects outward from the bacterial surface, essentially covering the cell wall like a dense forest.
And its primary function is molecular camouflage, right? I was reading about how it interacts with our complement system.
Yeah, this is where it gets really devious.
Because normally our complement system acts like a biological tagging mechanism. It floats around in our blood and slaps a chemical destroy me tag on foreign invaders.
Right, so our white blood cells, the neutrophils, know to come eat them.
Right, phagocytosis.
Yeah.
But the M protein somehow hijacks that entire system.
It does. It actually binds to our body's own complement regulatory proteins. Now, these are the proteins that our own healthy cells use to say, hey, don't attack me, I belong here.
Like a VIP pass.
Exactly like a VIP pass. By grabbing those specific host proteins and draping them over itself, the M protein effectively disguises the bacterium as self.
That's terrifying.
It is. The neutrophil bumps into the strep, reads the stolen VIP pass, and just moves on. The bacteria effectively resists being eaten.
But, eventually the immune system does figure out the disguise. I mean, it recognizes the specific M protein, builds antibodies, and clears the throat infection.
Oh, eventually.
So if we catch it, why do we get sick again the very next year?
Because the M gene is incredibly hypervariable. There isn't just one M protein structure. The CDC has actually cataloged over 220 distinct M types.
Wow. So it's basically a criminal with a wardrobe of 220 different hyper-realistic silicone masks.
That is the perfect analogy. The immune system spends 2 weeks learning to identify the first mask, catches the guy, and files the mugshot away.
But the next time the bug enters your body,
It's wearing mask number 47.
and biological memory is completely useless against the new disguise.
Utterly useless. Immunity to one M type provides absolutely zero cross-protection against the other 219.
Which explains why repeat strep infections are practically a standard feature of human childhood.
Exactly. It isn't an immune failure on our part. It's an evolutionary arms race where the bacteria just constantly change their physical profile.
And modern epidemiologists track these masks in real time, right? Kind of similar to how we track flu strains or COVID variants.
They do. Molecular epidemiologists rely heavily on M typing to map outbreaks. A stark example of this happened during the winter of 2022 to 2023 in the UK and Europe.
Oh, the pediatric surge.
Yes, there was a sudden, deeply concerning surge in invasive pediatric group A strep, and by sequencing the M genes, public health officials pinpointed that the outbreak was driven by a specific sublineage called M1UK.
And that variant was worse.
Much worse, it had acquired mutations allowing it to produce dangerously high levels of toxins.
Which brings us to a really dark reality about the M protein. We've established that it hides the bacteria. But when the immune system finally sees through the disguise and builds antibodies to attack it, that process can trigger a devastating chain reaction,
Yeah, we're stepping into the territory of acute rheumatic fever.
which our sources describe as one of the most tragic mechanical errors in human biology.
It really is.
Let's walk through the mechanics of this carefully because it's completely counterintuitive. Rheumatic fever strikes 2 to 3 weeks after a strep throat infection that was left untreated.
Right.
But the disease itself is not an infection of the heart. The bacteria aren't chewing on the heart valves. In fact, by the time rheumatic fever begins, the group A strep is usually entirely gone from the body.
The pathogen has completely left the building, but it leaves behind this massive case of mistaken identity, which we call molecular mimicry.
Right, the friendly fire.
Exactly. Our immune system generates antibodies targeting that coiled coil structure of the M protein. But structurally, on a molecular level, that alpha helical coil of the M protein looks virtually identical to proteins found naturally in human cardiac tissue.
Human myosin and tropomyosin.
Yes. So to a circulating antibody, the strep mask and the heart muscle look like the exact same target.
So the immune system, which is fully mobilized to hunt down the remnants of the strep infection, accidentally cross-reacts with the patient's own tissue.
It mounts an inflammatory attack on the heart valves, leading to permanent scarring. But it doesn't even stop at the heart.
Right, it attacks the joints too, right?
Yes, causing severe arthritis, and even the basal ganglia in the brain.
Which causes synonyms chorea, those involuntary movement disorders.
Just that.
The very antibodies designed to save you from a throat infection end up causing a multi-system autoimmune breakdown. Which, honestly, completely reframes how I think about a basic doctor's visit.
How so?
Well, when I get strep throat, I assume the doctor gives me a 10-day course of penicillin purely to make the pain stop faster.
Yeah, that's a huge misconception. Antibiotics might shave, a day or two off the symptomatic phase, but symptom relief is really a secondary benefit.
Okay, so what's the primary goal?
The primary clinical mandate for prescribing a full course of penicillin for a mild strep throat is to eradicate the organism rapidly and completely before your immune system has time to generate those cross-reactive antibodies.
Wow, so we are taking the drugs to protect our heart valves, not to soothe our throats.
We are intervening to prevent the molecular mimicry cascade from ever initiating.
That makes the global data on this incredibly sobering. Our sources point out a really stark health equity issue here.
Very much so.
Because in high-income nations where you can easily get a rapid test and, a cheap course of amoxicillin, rheumatic heart disease has become incredibly rare.
Almost unheard of in some places.
But in regions like Sub-Saharan Africa, South Asia, and the Pacific Islands where basic antibiotic access is limited, this preventable autoimmune complication remains a leading cause of cardiovascular death. We are talking about tens of millions of people living with severe heart failure.
All originating from an untreated childhood sore throat.
It is a devastating reflection of healthcare disparity.
It really is. The organism acts exactly the same everywhere. The only variable is early access to an inexpensive generic antibiotic.
Now, rheumatic fever is this slow, tragic, delayed mistake, but group A strep doesn't always play the long game. Sometimes it bypasses the throat altogether and launches a rapid, overwhelming systemic assault.
Right. Historically, clinicians categorize this bug neatly into superficial infections like throat and skin, and then those delayed autoimmune complications. But that boundary really shattered in the 1980s and 90s.
When hospitals started seeing a surge of aggressive, invasive syndromes.
Yes, most notably necrotizing fasciitis and streptococcal toxic shock syndrome, or STSS.
Okay, I want to tackle the tabloid panic from the 90s here, because the media just ran wild with the phrase flesh-eating bacteria.
Oh, they loved that phrase.
It sounded like an exotic, localized outbreak of a brand new predatory species.
The media narrative was completely sensationalized, but, the clinical reality was still terrifying. The thing is, there is no unique, standalone species of flesh-eating bacteria.
It's the exact same bug.
It is the exact same Streptococcus pyogenes that causes a basic strep throat. The difference just lies in the specific virulence factors the strain possesses, often M1 or M3 types, and its portal of entry.
So if it bypasses the respiratory mucosa and enters deep tissue through a laceration or, say, a surgical wound,
it behaves entirely differently.
And it causes shock and organ failure in a matter of hours. I was reading about how it pulls this off, and it relies on these biological weapons called superantigens.
Superantigens, yes.
And to understand a superantigen, we really have to look at normal immune activation first. Normally, if a pathogen gets into a wound, an antigen-presenting cell shows a piece of the invader to your T cells.
The generals of the immune system.
Right. And that normal antigen might activate roughly 1 in 10,000 T cells. It's a highly coordinated, targeted strike force.
A measured response, perfectly tailored to the specific threat. But Group A strep deploys superantigens like SpA, which, notably, is the exact same toxin responsible for the red rash and scarlet fever.
Oh, interesting. So what does the superantigen do?
Well, a superantigen doesn't wait to be processed normally. It acts as a crude chemical bridge. It forcibly cross-links the receptors on the antigen-presenting cell and the T cell, literally locking them together.
So instead of a targeted strike, what happens?
That forced connection bypasses all the normal regulatory checkpoints. Instead of activating 1 in 10,000 T cells, a single superantigen can simultaneously activate up to 30% of your entire T cell population.
3%.
All at once.
That's, okay, it's like a normal antigen is someone pulling a fire alarm in one building because a microwave caught on fire.
Yeah.
The local fire department shows up, handles it, goes home.
Right.
But a superantigen is like a hacker infiltrating a municipal grid and simultaneously triggering every single fire alarm, sprinkler system, air raid siren, and emergency broadcast in the entire country at the exact same second.
Yes. And the result of that systemic panic is total infrastructural collapse. In human biology, we call this a cytokine storm.
A cytokine storm.
Exactly. The wildly overactivated T cells dump massive quantities of inflammatory messenger proteins, cytokines like TNF-alpha, IL-1, and IL-6, directly into the bloodstream.
And normally those cytokines just tell local blood vessels to dilate slightly
Right.
to let immune cells reach an infection.
Usually, yes. But when released systemically at this insane volume, they cause catastrophic capillary leak.
The blood vessels literally lose their structural integrity.
Fluid leaks out of the circulatory system into the surrounding tissues. Blood pressure just bottoms out into severe hypotensive shock, and the major organs stop receiving oxygenated blood.
So the immune system's hyperreaction actually becomes the mechanism of death.
The bacteria effectively use our own defense mechanisms to destroy our cardiovascular stability.
Man. So when faced with an organism capable of all this, I mean, the 220 masks, the molecular mimicry, the superantigen hacker attack, how do we actually fight it? The answer in our sources really surprised me because it relies on penicillin.
It is one of the most remarkable anomalies in all of microbiology. After nearly a century of intense global usage, Streptococcus pyogenes has never developed clinically significant resistance to penicillin.
Never.
Never. Unlike Staphylococcus aureus or pneumococcus, which continually mutate to evade our drugs, penicillin still kills Group A strep just as effectively today as it did in the 1940s.
Which is fantastic for strep throat, but I have a mechanical question about treating the invasive stuff.
Sure, go ahead.
Penicillin kills bacteria by inhibiting cell wall synthesis, right? It essentially ruins the structural bricks, right as the bacteria are trying to build new houses during replication.
Exactly. It targets dividing cells.
So if I have a patient with massive necrotizing fasciitis, their tissue is packed with bacteria. But if penicillin works by attacking the replication process, throwing penicillin at a dense infection where the bacteria might not be actively dividing, shouldn't that fail?
You just described the eagle effect.
The eagle effect.
Yes, it's a critical caveat in infectious disease medicine. In a severe, deep tissue infection, the bacterial load is so incredibly dense that they exhaust all the local nutrients. To conserve energy, they enter what we call a stationary phase.
So they stop building new cell walls.
They stop dividing entirely.
So they are essentially sitting in the construction trailer, drinking coffee, doing no actual building, but still pumping toxic fumes into the neighborhood.
That's a great way to look at it.
And tossing defective bricks at a construction crew that has stopped working accomplishes nothing. The penicillin is present, but it has no active target.
Precisely. The bacteria just hunker down, completely ignoring the penicillin while continuing to manufacture and release those deadly superantigens.
Wow.
This is why giving high-dose IV penicillin alone for toxic shock syndrome is totally insufficient. Clinicians must add a second antibiotic to the regimen, clindamycin.
Okay, and clindamycin doesn't target the cell wall. It targets the ribosomes, right? It inhibits bacterial protein synthesis.
Exactly. It shuts down the bacteria's ability to manufacture proteins. By adding clindamycin, you aren't just hitting the bacteria harder, you are specifically turning off the toxin factories.
Ah,
Even if the bacteria are in a stationary phase and completely ignoring the penicillin, clindamycin enters the cell and forcibly stops the production of superantigens like Sp.
you literally cut the power to the trailer so they can't broadcast the hacker signal anymore.
Perfect analogy.
That principle, that the specific mechanism of the antibiotic is just as critical as its overall effectiveness against a bug, is just a masterclass in clinical logic.
It highlights exactly why treating infectious disease requires understanding the dynamic state of the organism, not just reading a basic susceptibility chart.
So to pull all these threads together,
Hmm.
we've tracked a single microscopic organism through a staggering spectrum of disease today.
We covered a lot of ground.
We started with Rebecca Lansfield using hot acid in the 1930s to find its chemical fingerprint. We unmasked the M protein, exploring how its 220 disguises exploit our complement system. We broke down the tragic molecular mimicry where our own antibodies scar our heart valves.
Right.
And we examined the chaotic cytokine storms triggered by superantigens and how clindamycin is used as a targeted strike on those toxin factories.
It truly illustrates how much systemic devastation a pathogen can achieve. And not necessarily by overwhelming us with sheer numbers, but by elegantly manipulating our own biological rules against us.
And as we wrap up this deep dive, there is one final, incredibly provocative detail hidden in the research.
Oh, this is my favorite part.
We know that Streptococcus pyogenes is incredibly dangerous, but it turns out it doesn't naturally possess all of its worst weapons.
No, it doesn't.
It often acquires the blueprints for its most lethal superantigens, like Sp, when the bacterium itself gets infected by a bacteriophage.
A virus that strictly infects bacteria.
Right. The bacterium catches a viral infection, and the virus's genetic material gifts the strep a deadly new superpower.
It's an incredible irony.
Its evolutionary leaps literally rely on these viral phages, which raises just a fascinating question. If bacteriophages are the very vehicles that manipulate and weaponize this bacteria, could the future of treating antibiotic-resistant superbugs, or even these severe invasive strep infections, lie in phage therapy?
It's a huge area of research right now.
Could we eventually isolate those exact same viruses, strip out the toxin genes, and deploy them as biological assassins to hunt down the strep?
Turning their own evolutionary mechanics into a precision weapon. It really is one of the most promising frontiers in modern microbiology.
It's just something to mull over next time you're staring in the mirror, wondering why your immune system just can't seem to remember the throat bug it fought off last year. Thank you for joining us on this deep dive into the microscopic world. Stay curious.